Good morning, all. So, today we are going to start our topic, which is photosynthesis. Before starting the topic, I would like to show you some videos regarding photosynthesis so as to clear the concepts. What is chloroplast? What are stomata? Inside the leaf, there are stomata, chlorophyll, and where are they located? All those can be seen first, and after that, I will be showing you how to I will be explaining you how the photosynthesis is related to that of photochemistry. That is, the photosynthesis is a naturally occurring photochemistry, and that can be utilized in the preparation of solar cells. So the idea behind solar cell was originated from photosynthesis. So today we are going to start our topic, which is photosynthesis. Let's begin with some videos. So this is the first video. It will explain you what is photosynthesis.
Hey there! It's a lovely sunny day today. So I came down for a sun bath. Don't look at me like that. Didn't your teachers tell you that sunlight is a good source of vitamin D? Hey! Don't forget that we trees and plants make our food from sunlight. Yes, you're right, Mr. Tree. Isn't the process called photosynthesis? Yeah. Come friends, let's learn about photosynthesis today. Zoom in. Photosynthesis. Doesn't that sound like a big word? Well, don't be bogged down by it. I'll tell you what it means. Photo is a Greek word for light, and synthesis is a Greek word for putting together. Now, it's simple. Photosynthesis is using light to put things together. Plants use this process to make their food with the help of sunlight, water, and carbon dioxide. Did you know that plants breathe just like us? You didn't? Well, now you do. Plants have tiny openings called the stomata present in their leaves, through which they take in carbon dioxide. Yes, they breathe in carbon dioxide and give out oxygen. They also use water and other nutrients to make food which is absorbed by their roots. The leaves contain tiny pigments called the chloroplasts. These pigments take in carbon dioxide, water, and sunlight and turn them into sugar and oxygen. The sugar is then used by the plants as their food, and the oxygen is given out into the atmosphere. This process as a whole is called photosynthesis. Am I right, Mr. Tree? Yes, you are. Trivia time! Chlorophyll is a green pigment that is found in the chloroplast of the plant. It makes the plant look green. Plants are often known as lungs of the world as they take in carbon dioxide and give out oxygen, which the humans breathe. It's time for some more sun bath. So this is me zooming out. Tune in next time for some more fun facts.
So, myself, Swati Villekar, and I'm going to start the topic today, which is photosynthesis. So, basically, you got the idea about photosynthesis. What is it? It is nothing but the combination of CO2 and water in the presence of light and formation of carbohydrate along with the liberation of oxygen. So, this is the main reaction, that is CO2 combining with water, producing glucose as well as oxygen with the removal of ATP molecules. So this So this is another video. Just see here, leaves are having tiny pores which are called as stomata, through which the oxygen will be removed. And CO2 enters the plant. Now this is the chloroplast, and inside it these are called as stroma part. Complete of this is not actually, this is called as granum, and each disc like structure is called as thylakoid membrane, and the other part is called as the stroma part. We will see that in one slide. So you are seeing what is photosynthesis. Photo means nothing but light, and synthesis means synthesizing something. So in the presence of light, CO2 from air and water from the soil combines together to produce carbohydrate, that is C6H12O6, and liberation of oxygen is there. Now this is the chart I have made you. It is the syllabus for same for where photosynthesis will be divided into two parts, which is the photophysical kinetics of biomolecular reaction, and the another one is solar cells. So the basic for the solar cell is the process of photosynthesis. So these solar cells are of two types. That is, in the solar cells there are two subpoints, which is solar energy conversion and storage, which will include photogalvanic cells and photovoltaic cells. So photoelectrochemical cell is there, and the base for all that is from photosynthesis. So this is the basic for same for also. So study with complete understanding. Now where the photosynthesis takes place. If you have this question, we all know that the light is being absorbed inside the leaf part of the plant, and it contains chlorophyll molecule where the photosynthesis takes place. Now this is the chloroplast. It is a three-dimensional chloroplast membrane, model of chloroplast membrane. This is the light dependent reaction which takes place in the thylakoid disc, and another one is light independent reaction which takes place in the stroma part. Now you can see the red or orange color portion, it is called as the stroma, which is the aqueous space. And these structures are called as complete to stack. The its complete stack is called as granum. It is the stack of thylakoids. So one of the disc-like structure, you can see it is a disc-like structure, which are here they are having 1, 2, 3, 4, 5, 6 discs in this picture. So each disc is called as separately thylakoid, and the stack of this thylakoid is called as the granum. So this is the outer membrane for chloroplast, and this is the inner membrane for chloroplast, and the open space is called as the stroma part. So the light dependent reaction will be takes place in the thylakoid membrane, and light independent reaction will take place in the stroma part. So light dependent reaction includes the photosystem 1 and photosystem 2, where there occurs the generation of ATP and NADPH, which are the energy storing molecules, and those are used in the stroma part for Kelvin cycle, where CO2 will be converted to carbohydrate. So we will see now photosystem 1 and photosystem 2. These are the photosynthetic cycles and the reaction centers, which is called as photosystem 1 and photosystem 2. So this is photosystem 1. As you can see, this is the photosystem 1 and this is the photosystem 2. Sorry, this is the photosystem 2. Here it is mentioned as 2. This is the photosystem 2 and its reaction center for photosystem 2 is P680. And the another one is photosystem 1, for which the reaction center is P700. Now we will see here, this is the chlorophyll B molecule and this is the chlorophyll A. Here there are chlorophyll A, chlorophyll B, here there is chlorophyll A molecule. Now, what is the main reaction between PS1 and PS2? In PS1, firstly the water will get split into hydrogen and oxygen, and there occurs the liberation of electron, and those electrons are trapped by this center, that is P680, that is PS2, and it requires, so if there are two hydrogens, if there are two water molecules, they will liberate the four electrons. We will see that in the reaction. So the four electrons which are being liberated are trapped by the PS2 center. So this is the trap where the electrons are being trapped. Then they will move towards the plastoquinone. From plastoquinone, they will move towards the cytochrome M. But during this, there occurs the generation of four ATP molecules. Through cytochrome M, the electrons will move towards the plastocyanine. From plastocyanine, they will reach the photosystem 1, which is the P700 center of the chlorophyll A molecule. From that, the electrons will be trapped in that on the absorption of some energy. Here also there is absorption of energy, and here also there is absorption of energy, and those four electrons are being trapped in that center, and then they will be transferred to the bound ferridoxine iron sulfur protein, and from that iron sulfur protein they will be transferred, electrons will be transferred towards the ferridoxine. From ferridoxine they will be transferred towards the NADP, where there occurs the formation of NADPH molecule. So this NADPH molecule and ATP molecule, these are being liberated during this PS1 and PS2 cycle. So these are being used for the reduction of CO2 to carbohydrate during the Kelvin cycle. And here, after this, we will see the Kelvin cycle. But before that, we should know the reduction potential. That is the redox potential for PS2. It is about 0.8, and that for redox potential for PS1 it is about 0.4. So just remember it, the reduction potential for PS2, redox potential for PS2 is 0.8, and that for PS1 it is 0.4. So now we will see the Kelvin cycle. In Kelvin cycle, you can get the better pictures for Kelvin cycle. Here, the CO2 which has been removed here the CO, the NADPH molecule and ATP molecule, they will be used for the reduction of CO2 to carbohydrate, and CO2 is coming from the air through stomata. So when CO2 is entering through the stomata, at that time, if there are three molecules of CO2, then it will produce the 6,6 carbon unstable compound, which will be converted to phosphoglyceric acid, which is again having, as there are three molecules of C6, it will produce one molecule will break into two molecules of C3. So if it is one molecule, then you will see, if it is a one molecule of C6, it will be broken down into two molecules of C3. Likewise, as there are three molecules of C6, it will be broken down into six molecules of C3. So firstly, the phosphoglyceric acid will be formed through, then after phosphorylation, that ATP is being utilized here, and it will again be converted into ADP. As you all know, ATP is the energy storing energy storing molecule, and it is having, when it combines with the inorganic, when it breaks down, it gets broken down into ATP plus inorganic phosphate with the removal of energy, and that phosphate is being utilized here. So phosphoglyceric acid combines with the phosphate from ATP and generates the C3 phosphate. Then this phosphate again undergo reduction and produce the phosphoglyceraldehyde by using the NADPH molecule, and it will itself get converted to NADP. Now this phosphoglyceraldehyde has the possibility that it will be the one molecule of phosphoglyceraldehyde may undergo the glucose synthesis, where there occurs the formation of glucose, and the other molecules of phosphoglyceraldehyde forms the ribulose monophosphate, and then as it is monophosphate, it will again undergo phosphorylation, and it will produce the mono diphosphate from monophosphate by the absorption of inorganic phosphate here, and in this way this ribulose diphosphate, which is having a C5 carbon, when combines with CO2, at that time one carbon will increase there and it will produce the C6 entity. And so that's why they have taken here three molecules of C6. Now, every turn of the cycle fixes one molecule of CO2 by producing two molecules of PGA. It means if one molecule of, as they have given the ratio 3 and 6 molecules of phosphoglyceraldehyde are being here generated. So here, what are they saying? Every turn of the cycle fixes one molecule of CO2 by producing two molecules of phosphoglyceraldehyde, and then two molecules of firstly for one molecule of phosphoglyceric acid and then two molecules of phosphoglyceraldehyde. The three turns are necessary to release one molecule of phosphoglyceraldehyde, that is C3, for the glucose pathway for the remaining 5 PGAL molecule remaining within the cycle, and thus 6 turns produce sufficient quantity of phosphoglyceraldehyde for the production of one molecule of glucose, as it is C3 plus C3, that is C6. Glucose will be C6 H12O6. So when one molecule here combined, at that time it will require the 6 cycles, because each molecule is generating the 6 turns produce sufficient quantities of phosphoglyceraldehyde for the production of one molecule of glucose. So they will produce sufficient, 6 turn will produce sufficient quantity of phosphoglyceraldehyde which will be utilized for the production of glucose. And this reaction is taking part in the stroma part, which is the dark reaction. Again, we will just revise it. What is that? The CO2 from air comes towards the plant through stomata, and then it combines with ribulose diphosphate, which is a C5 sugar, and it gets converted to 6 sugar, which is a stable compound. Now this will be converted into phosphoglyceric acid. From phosphoglyceric acid, it will be undergoing phosphorylation, where it will form the phosphate group with the use of ATP, and then that phosphate entity will undergo reduction and produce the phosphoglyceraldehyde, and then one of the molecule of phosphoglyceraldehyde from 6 molecules will undergo the glucose synthesis, and the remaining 5 molecules of phosphoglyceraldehyde will undergo formation of ribulose monophosphate, and from the ribulose monophosphate they will again undergo phosphorylation and form the ribulose diphosphate. In this way, there occurs the use of 9 ATP molecules and 6 NADPH molecules during one cycle of Kelvin cycle. Now, as we have seen this photosystem 1 and photosystem 2, how they are actually taking place inside the membrane, that we are going to see. As we have seen the thylakoid membrane. So this is the thylakoid membrane, which is having some bilayer lipid structure. It is comprising of PS2, then PS1. PS2 is nothing but P680 center, then plastoquinone, cytochrome F, plastocyanin, then PS1 center, ferredoxin, iron sulfur protein will be there, and the generation of ATP molecules will be there. So what we have seen there, initially water from the soil which is being coming towards the plant through stems, through roots. That water will undergo liberation, producing the water as well as H plus ions. Now, these H plus ions will be utilized for the reduction of NADP to NADPH. That NADP will be reduced to NADPH by the use of these protons, which are being generated through the photolysis of water. Then you can see to the now what happens at the same time, 4 electrons are being generated during this splitting of water, and those 4 electrons are trapped inside the reaction center P680 on the absorption of some light radiation, and those electrons which are being shown here by the blue arrows, these are transferred to the plastoquinone, and that plastoquinone transfers the same to the cytochrome F. Through the cytochrome F, electron will be transferred to plastocyanine. But during this journey, there occurs the generation of ATP molecules, and those ATP molecules will be formed as ADP, combining with inorganic phosphate and generating the ATP. So the process will be occurring here. Now, the electrons which are being moved towards the plastocyanine, they will move towards the PS1 center, which is P700 center, and it will go to the iron sulfur protein. From that iron sulfur protein, it will reach to the ferredoxin, and then after ferredoxin, there occurs the generation of NADP to NADPH, and this NADPH and ATP are utilized in the Kelvin cycle in the stroma part. So this is the structure for thylakoid membrane. So here the same I have taken, but it is different picturization. Firstly, this is the P680 center. It will absorb some amount of energy and then go to the excited state where it will move towards the plastoquinone. From plastoquinone, it will move towards the cytochrome F, where there occurs the generation of ATP. From that, it will go to the plastocyanine. Then it will move to the P700 center. The electron are being moving in this way. Then from P700, it will absorb the amount of energy, and again it gets excited, and the electron will be transferred towards the, which are shown in red arrows. The electron are being transferring towards the iron sulfur proteins. From iron sulfur, it will be transferring towards the ferredoxin. Then from ferredoxin, it will be converting NADP into NADPH. And the enzyme for that is ferredoxin NADP reductase. So one more video which will give you the clear idea about the what is Kelvin cycle, what is photosystem 1, what is photosystem 2. It is a 7 minutes video. So watch it very clearly, because photosynthesis occurs when the energy is being utilized from the visible region of the spectrum. As we know, the white spectra, from that white spectra, the photosynthesis takes place when the light is being absorbed from the visible region, and after the trap of light, there occurs the photosystem 1, photosystem 2, which are the energy generating steps in the thylakoid membrane, and from the thylakoid membrane, the CO2 that CO2 will be converted to carbohydrate in the stroma part. So this will all be given in this video. So watch it. After watching it, if you get the clear idea about the video, then you can easily understand the concept behind the photochemistry behind the photosynthesis.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the Earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of two sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. While the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life, providing food and oxygen for the complex food webs on both land and in the oceans.
Now we are going to start our content of the topic, which is photosynthesis. In a broad sense, photosynthesis in plants is a photo-induced, means the PS2 center is force absorbing the light and it is transferring the electron to the PS1, and PS1 also that it is absorbing light and it is transferring the electron and converting NADP to NADPH. So photosynthesis in plants is a photo-induced electron transfer reaction. Chlorophyll molecules in the green plants are the main light harvesting molecules. They are assisted by carotenoids and photocyanins in this act. As we have seen in the photosystem 1 and photosystem 2, there is photocyanins are there. The electron are being transferred to the photocyanins first. This is the plastocyanin. The chain is being plastoquinone, cytochrome F, then plastocyanin. So they are assisted by carotenoids and photocyanins in this act. These molecules have absorption in the visible region, covering the whole spectrum from blue to red. As we are saying it, VIBGYOR, it is only absorbing the radiation from blue to red. Then the energy absorbed by these molecules is transferred to chlorophyll A, which is that chlorophyll A means whatever the energy is being absorbed, it is transferred from chlorophyll PS2 center to PS1 center, and PS1 center is comprising of chlorophyll A. So that chlorophyll A is the main light sensitive molecule. So the chlorophyll B will also transfer its energy to chlorophyll A. As here we have seen, these are the this is the chlorophyll B, it is having a P680 center, and this is the chlorophyll A. So chlorophyll B is also giving its energy to chlorophyll A. So chlorophyll B is also transferring its energy to chlorophyll A, which has two absorption peaks. That chlorophyll B is having the two absorption peaks, one in the blue violet region where the wavelength is 448 nanometer, and another in the red region having the wavelength of 680. It means in vitro it emits the red fluorescence with a wavelength at 680 nanometer, and this characteristic fluorescence, that is which is emitting the red fluorescence at it as it is being giving out the red light. So this characteristic fluorescence is the only fluorescence observed. Whichever is the light absorbing molecule, this indicates that the other molecules transfer the excitation energy to chlorophyll A, and The photosynthetic process utilizes the low energy quanta corresponding to red light only. In vitro, the fluorescence efficiency of chlorophyll A is about nearly 0.3, and in vivo, this quantum efficiency of fluorescence is much more reduced, and any factor which will reduce the fluorescence enhance that will enhance the photosynthesis, or we can say any factor which will enhance the fluorescence that will reduce the photosynthesis. So this is the chlorophyll molecule. It's having a structure. Just you will have to try to draw the structure. Firstly, there will be magnesium in between, and this magnesium is joined to the four side by the, as you can see, first, second, there will be the four furan rings will be there. So not furan, there will be the pyrrole rings where the rings will be there, and this is the pyrrole ring structure. 1, 2, 3, and 4. There are four pyrrole rings are there in the chlorophyll structure, and they are joined together through magnesium and a long chain, and this is the long chain. Just remember the structure because we want it in the next slides. So there are four pyrrole rings are there, and in between the four pyrrole rings there is magnesium, and these are joined together. They are being connected to one another, and one of the ring is being joined to a long carbon chain, where if this R group is CH3, then it is called as the chlorophyll A molecule, and if R group is CHO, then it is called as the chlorophyll B molecule. It depends on this R. If it is CH3, then it is chlorophyll A. If it is CHO, then it is chlorophyll B. And this is the absorption and emission spectra for the different chlorophyll molecules. So this is the chlorophyll molecules and absorption spectra of the pigments. So what different pigments are: chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, bacteriochlorophyll, and sunlight. So you can observe it. Chlorophyll A, for that chlorophyll A they have given these lines. Chlorophyll B, I think this is for chlorophyll B. Then straight line is there for chlorophyll C. This is the straight line for chlorophyll C. Then dotted structure like this, chlorophyll D. Then bacteriochlorophyll, this structure is for bacteriochlorophyll. It will be having the absorption in the range of 780 near about. And then for sunlight they have given these lines. So through the absorption peaks we can know where it is having the maximum wavelength, so that it will be the, then we can see it is a PS1 center. As we are saying, the PS1 center is having a wavelength of, PS2 center is having a wavelength of 680, PS1 is having a wavelength of 700. So from this absorption peak we can say that. Now the net reaction of photosynthetic process in plants can be given as n times CO2 combining with n times water producing we can say 6 molecules of CO2 combining with 6 molecules of water producing carbohydrate and 6 molecules of O2 at the reaction center of first by the absorption of light, and this is 680 PS2. Now the energy which is equivalent of red light is about 190 kilojoule, but the actual energy requirement for reduction of 1 mole of CO2 to that of carbohydrate is about 470 kilojoule. Means the mechanism of reduction of CO2 is much more complex. So the initial act of electron transfer process from photoexcited chlorophyll A, water serves as an oxidizable substrate producing O2. As we have seen in first slide, we have seen that water is being split into oxygen and H plus ions. H plus ions are being trapped by the NADP and they changes to NADPH, and the oxygen is liberated, oxygen which is being liberated through the breaking of water is being coming out through stomata and going to the nature, and also as an electron source for the reduction of CO2. So the water is the electron source as well as the oxygen source. So the two quanta are required to create one oxidized and one reduced species, that is for the transfer of one electron two quantas are being required. For one electron, it requires two quanta. Means for 4 electrons, it will require the 8 electrons. So for evolution of one molecule of O2, 4 electron or 8 quanta of light will be required. So this will be the process for water, and this question is asked in exam that prove that how 8 quanta are being required for the evolution of one molecule of O2. So you just have to write all this. So twice water molecules will be broken down into 4 H plus ions and 4 electron and O2. So as one electron requires the 2 quanta, so 4 electrons will require the 8 quanta. So the reduction of carbohydrate CO2 to carbohydrate is a dark reaction which occurs in a number of enzymatic steps in Kelvin cycle. Thus, photosynthesis, it is a cooperative process in which light quanta are pulled in the reaction center by various light harvesting pigments. From quantitative studies, it has been established that a set of 3000 molecules of chlorophyll A could initiate the chemistry needed for the evolution of one molecule of oxygen. If one quantum is responsible for one photochemical act, it can be estimated that a single quantum absorbed in a set of 3000 by 8, or we can say 400 chlorophyll A molecule causes just one oxidation reduction event in the reaction center. Thus, the concept of photosynthetic unit can be defined as one in which about 400 molecules of chlorophyll serves as one reaction center. It means the 400 molecules of chlorophyll that serves as one reaction center, as you have seen it in the video of 7 minutes. The chlorophyll molecule in the reaction center differ in the environment from the light harvesting chlorophylls and hence differ in the absorption wavelength. It is 700 nanometer in green plants and 870 nanometer in bacteriochlorophyll, which are designated as P700 center and P870 center respectively. Means the centers are being recognized through the absorption wavelength. Now, from a study of wavelength dependence of photosynthesis, immersion observed that the efficiency dropped in the red region of the spectrum. But if the system was irradiated with the blue and red light simultaneously, then the efficiency was regained. These observations led to the idea that two photosystems are involved in photosynthesis, and they are termed as photosystem 1 and photosystem 2. So the what immersion observed that efficiency dropped in the red region, but if the system again irradiated with the blue and red light, then that efficiency was regained. As the efficiency was regained on the absorption from the blue and red light, it confirms that there are two photosystems which can be termed as PS1 and PS2. A cooperation between these two systems is necessary for efficient working of the photosynthetic cycle. That is, photoexcitation in PS2 creates a strong oxidant and a weak reductant. We have seen that PS2, it is having a redox potential of 0.8. So it is a strong oxidant and weak reductant. PS2, and the strong oxidant, that is this strong oxidant is of the nature of quinone. That's why we say plastoquinone there. So it is of the nature of quinone and accepts the electron from water to liberate O2. A binuclear manganese complex is supposed to act as a catalyst in the oxygen evolution process. So the redox potential of the oxidant is between 0.0 to 0.12. Thus, a potential gradient of 0.6 to 0.8 volt is created there. Since the A0 value at PS7 for the couple of oxygen to water is 0.8 volt. The trapped P700 in PS1 is a special kind of chlorophyll A and is present in the ratio of 1:300 in the reaction center, and its redox potential is 0.4 volt. In the primary photoact in PS1, strong reductant, here just opposite of this, here in PS2 center there was the strong oxidant and weak reductant, and here in this case there will be the strong reductant and weak oxidant, just opposite of that PS2. So in the primary photo act in PS1, the strong reductant and weak oxidants are formed. Now the weak reductant from PS2 and weak oxidant from PS1. They couple through a number of different cytochromes. As we have seen their cytochromes also, they get coupled through the cytochromes of graded redox potential ranging from 0.0 to 0.4 volt. As we have seen here, the photosystem 1 which is having the strong oxidant and weak reductant, and this is the photosystem 1 which is having the weak oxidant and strong reductant. So this combines through the cytochrome series, as well as plastoquinone is there. Now during the downhill electron transfer, as you can see the electron transfer from PS2 center to PS1, this is the downhill electron transfer. So during the downhill electron transfer over the enzymatic chain, some amount of energy is degraded simultaneously, storing a part of it as phosphate bond, that is ATP, through the reaction. As here, from plastoquinone to cytochrome F, there occurs the generation of ATP, and then after ferredoxin, there occurs the formation of NADPH. So during the downhill electron transfer over the enzymatic chain, some amount of energy is degraded and simultaneously storing a part of it as phosphate bond of ATP through the reaction ADP plus IP. Just remember what is IP? It is the inorganic phosphate that combines with the adenosine diphosphate and forms the adenosine triphosphate species. So about 30 to 34 kilojoule per mole is the energy is the stored. The strong reductant transfers an electron through a number of enzymatic steps to NADP to form NADPH, and the intermediate enzymes are complex of iron sulfur proteins such as ferredoxin and hydrogenase. So in the absence of oxygen or low concentration of CO2, hydrogenase can liberate the molecular hydrogen and thus reduced NADPH. Thus then utilizes the energy stored in high energy phosphate bond ATP for the reduction of CO2 to CH2O in a number of dark steps, which we have seen in the Kelvin cycle. So the sequence of reactions in which CO2 is first added to a 5-carbon sugar was illustrated by M. Kelvin and his associated, and it is being described by the Kelvin cycle. So this is the Kelvin cycle where we have seen the ribulose monophosphate combines with CO2 and forms a 6-carbon unstable sugar, and then it gets broken down into the 3 molecules gets broken down into 6 molecules of C3 and of phosphoglyceric acid. That utilizes the ATP molecule, gets converted to some phosphate, and through the phosphate that will undergo reduction by utilizing the NADPH and gets converted into phosphoglyceraldehyde, and there occurs the generation of 6 molecules of phosphoglyceraldehyde, out of which 1 molecule go into the glucose synthesis. But it requires, for the glucose synthesis, it requires the 6 molecules of phosphoglyceraldehyde, and the remaining molecules will go towards the formation of ribulose monophosphate, and that ribulose monophosphate again combines with the ATP and produce the ribulose diphosphate, which will again initiate the chain. And in this way 6 turns are needed because each turn is generating the 1 molecule. So in this way it will need 6 turns produce sufficient quantity of PGL for the production of 1 molecule of glucose. So photosynthesis in plants occurs in the thylakoid membrane, which we have already seen the thylakoid membrane. Structure of thylakoid membrane I have shown you. This is the structure of thylakoid membrane. Bilayer lipid structure is there. PS2 center, PS1 center, plastoquinone, cytochrome F, ATP, ferredoxin reduce FES. This is ferredoxin. This is the enzyme, that is ferredoxin NADP reductase enzyme is being there. So just remember all those in your mind. So photosynthesis in plants occurs in the thylakoid membrane, and these are the lamellar structures, organized stacks in the leaf chloroplast. There are regions of greater density which are called as the grana, as different from that of stroma. That is, when the thylakoids are being placed on one another, then the complete disc, complete structure, that is called as the granum. And there are lots of granum are there, so they are called as the grana. So these are the regions of greater density which are called as grana. They are different from that of the stroma. By the action of detergent on the isolated chloroplast, it has been demonstrated that PS1 and PS2 centers are located in different parts of the membrane, as we have seen in that picture. So chlorophyll molecules are the flat molecules with conjugated system of double bonds consisting of 4 pyrrole rings. So the chlorophyll molecules are flat molecules with a conjugated system of double bonds where they are consisting of 4 pyrrole rings which are joined in a circle and attached to a long hydrocarbon chain, which is the phytol chain. Through, through this long chain, the light absorbing molecule is attached to the lipoprotein of the membrane in regular ordered arrangement. So this was the structure of chlorophyll molecule, where the 4 pyrrole rings are being joined to one another in a circular way, and they are joined to a long chain of carbon. So through that long carbon chain, they are attached to the lipoproteins of the membrane in a regular ordered arrangement. So the membrane plays a very important role in imparting unidirectionality to the electron transport by separating oxidation and reduction centers. The membranes are composed of lipids, pigments, and protein complexes. They provide potential gradient for the transfer of electron across the membrane from PS2 to PS1. The coupling is not by chemical substances but by electrochemical states, and it has been pointed out that the primary quantum conversion apparatus in photosynthesis behaves in a similar way that of the photoconductive semiconductors, and in this way that separates the positive and negative charges as electrons and holes. So a physical separation of electron and positive hole or oxidant is brought about the membrane, preventing back recombination. So this is the membrane where water is being splitted, and this is the chlorophyll molecule which on absorption of light will undergo excitation, and on the absorption of electron it will become the negatively charged, and then it will give its excess electron to the acceptor species, as a result of which acceptor will become negatively charged and it will come to the ground state. But the back reaction is being prevented here. It will only go in the irreversible way. So thus in photosynthesis, the initial event are a light stimulated charge separation. A cooperation of a suitable electron acceptor and a donor increases the efficiency of charge separation at excited chlorophyll molecules. Under these conditions, these molecules act as photon-powered electron pump. As we have seen on the absorption of photon, electron is being transferred from PS2 to PS1. So these molecules act as photon-powered electron pump for the transfer of electron uphill from electronic levels of the donor to the electronic levels of the acceptor. So the oxidized chlorophyll molecule, that is chlorophyll with a positive hole, constitutes a strong oxidizing agent and should be capable of oxidizing compounds with fairly high redox, which is 0.8 volt. So the recombination of excited electrons and the hole left behind is called as an exciton. That is excited electron and a hole. So it is called as the exciton. They are prevented from recombination by the hydrophobic interior of the membrane. So the exciton migrates through the photosynthetic unit until it meets an oxidizing enzymatic center which serves as a trap for the excited electron. The exciton is now reduced to a free hole, which then migrates through a unit until it reaches an electron donating center. The primary photochemical oxidation reduction process is thus completed, where, as you can see here, the primary oxidation reduction process is being thus completed and the oxidation product being formed at one enzymatic center and the reduction product is being formed at the another center. So the chlorophyll itself is not decomposed but it only sensitizes the reaction. The light energy is stored in the form of chemical energy in the reduced form of the oxidant, NADPH. So the such ideas have initiated in vitro studies of chlorophyll asensitized reaction in photosynthetic membranes from the laws of electrochemical kinetics. It has been possible to mimic the energy conversion and storage capacity of these membranes by experimental bilayer lipid membranes formed from chloroplast lamella extracts. So a photoelectrochemical cell can be constructed by placing two redox couples of graded potentials on either side of the membrane and dipping metal electrodes in each of them. So on completing the circuit by suitable device, photocurrent will be generated on excitation of the membrane by light energy. Then the alternative sequence of reaction initiated by light can be summarized as follows, where the chlorophyll on the absorption of photon, it will form an exciton formation. There occurs the exciton formation. Electron hole pair will be there, which will combine with water and it will generate this species. Then all these reactions will be there. So here chlorophyll denotes a photosynthetic pigment plus minus pair. Chlorophyll plus minus pair is an electron hole pair or an exciton. And then E aqueous Q, this is E aqueous, it is a hydrated electron that is on the combining with water, it is forming the hydrated electron along with the generation of positive charge, that is hole. Then A minus and D plus, these are the charge transfer complexes with an electron acceptor and an electron donor. So chlorophyll plus and chlorophyll minus, that is A minus chlorophyll plus and D plus chlorophyll minus, these are the electron acceptor and electron donor complexes, charge transfer complexes of an electron. That is, I will read it again, that is A plus, that is chlorophyll plus, and D plus CH minus. So A plus, this is A minus CH plus and this is D plus CHL minus. These are the charge transfer complexes of electron acceptor and electron donor species respectively. And the last step is responsible for delayed emission of chloroplast. That is, as it is generating the some amount of light, so some amount of radiations are being given out there. So this is responsible for the delayed emission of the chloroplast, where chlorophyll minus combines with the chlorophyll plus and generates two chlorophyll molecules. You can study the reaction and you will have to practice this reaction. Just read it. And this is the membrane model for electron transfer reaction in the photosynthetic cycle with acceptor A and donor D. As you can see here, on the absorption of light, it will get excited, and here there is acceptor which will accept the electron, and there it is the donor, and this is the energy levels. You will, in this picture you will have to just talk it about the energy levels. The acceptor level is below that of the excited level, and donor level is below that of the acceptor level. So the energy difference between the acceptor and donor is called as the del G, which is the free energy. And that del G, which is nothing equals to NEF, is theoretically available electrochemical free energy of the electron and positive hole. So it has been postulated that complexes of electron transfer proteins in a membrane are of graduated redox potentials. The electron transfer occurs through channels provided by a complex sequence of ligands, and bonds are conjugated molecules like carotenoids. So these proteins are able to accept the electron from excited chlorophyll on one membrane side and donate them to an acceptor of more positive redox potential on the other side. So the membrane provides a resistance for an ion current from anode to cathode, which closes the electrochemical circuit and converts excitation energy into the chemical free energy del G. So these are the references from where I have taken the YouTube videos. So this is all about today's topic. Thank you. So this video is main. Thank you. I will end the broadcast and I will give one Google Meet link. Join that link.